So meiosis, and you're going to see that each of these points now basically contrast it to mitosis. So it involves two cell divisions as opposed to one, and it creates haploid instead of diploid, gametes instead of somatic cells. Don't worry, we're going to do a much more sort of focused comparison between mitosis and meiosis in a minute. The first meiosis occurs after chromosome duplication. So this parent cell is still going to go through interphase, which means it will still go through S phase. So its chromosomes will be duplicated. Meiosis 1, crucially, and this is important because you do get tested often on which chromosomes separate in which phases of the cell division. It separates homologous chromosomes into different cells. So this is my result of meiosis 1. And here are my homologous chromosomes separated. Chromosomes separated. Just really quickly, homologous chromosomes are chromosomes that have been inherited from a different parent, and that's why they've got different colors here, black and yellow. So, for instance, if this is chromosome 1, this is chromosome 1 paternal from the father, and this is chromosome 1 again, but inherited from the mother. When meiosis begins in the reproductive organs like the ovary and testes, it starts from a diploid cell, and every diploid cell contains two sets of chromosomes. Humans have two sets of 23 chromosomes, meaning I've got chromosome 1 from my mom, chromosome 1 from the father, chromosome 2 from the mother, chromosome 2 from the father, and so on. So here's now my homologous chromosomes separated. And then meiosis 2 finally will actually now go ahead and separate these sister chromatids from each other. And so this is meiosis 1, this is meiosis 2. And these are now my sister chromatids that are separated. Definitely make sure that your chromatids, definitely make sure that you're really comfortable with the terms homologous chromosomes versus sister chromatids and what they mean. Sister chromatids are genetically identical to each other and they're held together by the centromere. Homologous chromosomes are not identical to each other because they contain different alleles that were inherited. They've got the same genes in the same locations, but they've got different alleles because they were inherited by different parents. So the phases of both meiotic divisions, meiosis 1 and meiosis 2, are the same. It's prophase, metaphase, anaphase, telophase. And in terms of the general events, they have a lot of similarities with the phases of mitosis. So in prophase 1, chromosomes supercoil or condense, nuclear envelope breaks down, and spindles form, same as mitosis. What's the difference, though? That at the end of prophase 1, that homologous chromosomes are being separated in meiosis. In mitosis, only sister chromatids are separated, ever. In meiosis 1, it's homologous chromosomes that are separated from each other. So yes, all the mechanics are the same, nuclear envelope, chromosome condensation, mitotic spindle forming, but the types of chromosomes that are separated is what's different. And a really important event takes place at the end of prophase 1, which is crossing over, which is when sections of DNA are exchanged between homologous chromosomes, between the chromatids of homologous chromosomes. And so for AQA and Edexcel, you don't need to know the stages of meiosis. The reason is because they're very similar to the stages of mitosis. So if you learn the stages of mitosis, you're fine, and you just need to know how they differ from meiosis, basically. Meiosis 1, metaphase. So in metaphase, the chromosomes, if you can see here in this diagram that we've got regions of alternating like light blue and dark blue. So for instance, this chromosome here with the light blue might have been maternal chromosome. And this chromosome in the dark blue might have been paternal chromosome. And you can see that they've exchanged little portions of DNA between them. They now line up at the equator, and the spindle fibers are bound to this diagram should show you know that the dotted line is going directly onto the centromere. And this is where we have an important event called independent assortment or segregation. Both are correct. What does this refer to? So as we said, homologous chromosomes are not genetically identical to each other. They come from different parents. They contain different alleles. Which daughter cell, the position of them in the equator, determines which daughter cell is going to inherit which chromosome. Meaning what? These could have been flipped. So paternal chromosome could be going to this cell, and maternal chromosome could be going to this cell. Equally, this chromosome could be going on this side, this chromosome could be going on that side. This process, importantly, is random. It's a random process. And it introduces genetic variation. Why? Because the daughter cells are not going to be genetically identical, since they will each have a random mixture of paternal or maternal chromosomes. What then happens during anaphase and telophase? The chromosomes from each homologous pair are actually physically going to be pulled to the opposite poles of the cell, much like in mitosis. And then finally, at telophase, the chromosomes are going to start to uncoil, decondense, and the the nuclear envelope begins to reform around those two new nuclei. So here we have anaphase 1, and here we have telophase 1. The two nuclei that are temporarily formed each now contain one set each of the chromosomes. So we're going to see, we can see this pointing again in a minute. At this stage, at the end of meiosis 1, these daughter cells that are going to form after cytokinesis are already haploid because they only contain a single copy of the homologous chromosomes. Let's have a quick look at a question before we move on to cytokinesis. Before we move on to the meiosis 2. Mitosis and meiosis are two types of cell division, which produce somatic cells and gametes, respectively, in mammals. Respectively. State another difference between the cells formed by mitosis and meiosis. So one difference is that we were told that they contain, let's look at our context first. Our context is right here, comparing meiosis and mitosis. Our command word is state, no justification. It's only worth 1 mark as well. So brief, keeping it brief here. And my direction is to focus on the differences of cells produced. So what does that mean? You cannot mention just any difference between mitosis and meiosis, of which there are many. And this is why this question is only worth 1 mark. Otherwise, a comparison between mitosis and meiosis would be worth more marks, of course. But here we can only talk about the cells produced. So have a quick minute to get to think about the answer before I say it. So one difference is what type of cells are made, somatic versus gamete. And what's the other difference between those two cells? Somatic cells are diploid and gametes are haploid. So mitosis produces diploid cells, which all somatic cells are, whereas meiosis produces haploid cells. Just because it's one mark doesn't mean that you shouldn't write like a whole coherent sentence, right? Cells. Okay. Let's now move to the second meiotic division, which again is made up of prophase, metaphase, anaphase, and telophase. So, as we said, meiosis 1, before we start with meiosis 2, produces 2 haploid cells because there are no homologous chromosomes anymore to speak of. Maternal chromosome 1 from paternal chromosome 1 have gone to the 2 daughter cells. Doesn't matter which way around they went, but there are no homologous chromosomes anymore. So technically, if we define diploid and haploid with the respect of whether we've got sets of chromosomes in there, we are now at a haploid state. It doesn't matter that we've got sister chromatids still, because sister chromatids, so these are 2 haploid nuclei, or cells actually, even though they have sister chromatids still, which of course are going to be separated now in the second meiotic division, because sister chromatids are genetically identical to each other. So they're not considered homologous chromosomes to each other. And we define haploid or diploid by whether we've got homologous chromosomes or not. So now in prophase 2, what is going to happen? DNA will supercoil, nucleus disintegrates, spindle fibers form. Same as in any beginning of meiosis or cell division. Metaphase 2, the chromosomes are lined up at the equator of the cell. Now, while sister chromatids in mitosis are, generally speaking, genetically identical, sister chromatids here are not identical to each other. Why? Due to crossing over. So just remember this diagram here, which just shows it a bit more clearly, that the normally this sister chromatid, and this sister chromatid would be genetically identical, but because of crossing over, and crossing over, remember, introduces genetic variation because it creates new combinations of alleles. We're going to see that in a minute. This sister chromatid to this sister chromatid is not genetically identical. So why is that relevant for meiosis 2, for metaphase of meiosis 2? Because this basically introduces yet another round of independent assortment. So during anaphase, when the genetically different chromatids are separated to opposite poles, this leads to a second round of independent assortment or segregation, meaning what? Which sister chromatid ends up in which daughter cell means that I will have 2 nuclei that are, of course, not genetically identical. So the first independent assortment is meiosis 1, homologous chromosomes are separated. The second one is when those sister chromatids are separated, and the reason they're not genetically identical is because of crossing over. So we have now 4 genetically different and haploid cells where also the sister chromatids have gotten separated. Let's have a look at an exemplar question. Meiosis is used by organisms to produce gametes, such as sperm and egg cells. Explain why meiosis requires 2 stages of cell division. So, what is our context that we are talking about? Meiosis. That first sentence here. Oops. Context. Command. Explain. So, justify your choice in the context of this question. And then finally, direction. We have to describe what happens in these 2 stages of meiosis 1 and 2. Take a minute to structure and possibly write the question. Let's look at the answer together and break it down. So meiosis requires 2 stages in order to, 1, half the chromosome number. So this is one bit of crucial information where 1 mark goes to. What's the other thing that meiosis does? It separates the homologous pairs of chromosomes as well as sister chromatids. So that's my second mark. So those are basically, I'm describing the jobs of meiosis, right? It's required to separate homologous chromosomes and then sister chromatids, effectively halving the chromosome number. Let's look at crossing over. So we're going to talk about specifically how meiosis introduces genetic variation into sexually reproducing organisms and how that's an advantage of sexual reproduction over asexual reproduction. So what does crossing over do? We phrase it as, so first of all, it allows alleles to be swapped between homologous chromosomes. So as we said, homologous chromosomes are not genetically identical. They will have the same loci, meaning the same gene. So for example, in this position here, let me just use a different color. Sorry, guys, because you probably won't be able to see that really well. And it's important that you can write down the annotations if you need to. So I'm just going to go with red here. In this position, there will be a gene that controls, for instance, eye color, right? And I'll have the same gene in the same location, whether I am looking at the, let's say this is the paternal chromosome, let's say this is the maternal chromosome. So this individual has a diploid cell before meiosis takes place. So here we are before meiosis, before prophase 1, because it's during prophase 1, at the end of prophase 1, where crossing over takes place. So this is the alleles that are found on this chromosome. So this person is, for instance, homozygous for capital A, and they're also homozygous for another gene that codes for a different trait, gene B, that will be found in this particular location. So they are homozygous for uppercase B. The maternal chromosome, again, as we said, it's going to contain the same genes at the same locations, but different alleles because it came from another person. It came from a mother. So say that this person was homozygous here for lowercase b, and then they were also homozygous for lowercase a in this part of the chromosome. When these sections of chromosome swap between the other, so what happens is that the homologous chromosomes, they line up. They actually line up, or we can say pair up as well, because line up might make you think about metaphase, where they line up along the equator, whereas homologous chromosomes actually pair up. This is an important detail. They pair up during prophase 1, where they may not necessarily be exactly at the equator yet. Prophase 1. And at that stage, pieces of chromosomes literally intertwine, they twist around each other, and they physically exchange portions of chromosomes. So it causes these sections of chromatids to break off, and they're swapped to the homologous chromosome. Yes, they only involve one of the 2 sister chromatids, whichever one is closer to the other, but the sections that they exchange are between homologous chromosomes. So don't say that they exchange between sister chromatids. If this sister chromatid exchange a piece with this sister chromatid, I would never know because they're genetically identical. The sister chromatids are still genetically identical at this stage because they haven't undergone crossing over yet. So what can I see here? So this sister chromatid is going to be the same as before. It's going to be capital A, capital B down here. However, here it has exchanged. Sorry, I should have drawn the A a little bit lower. I just noticed. But it has exchanged that piece of chromosome. So now it's going to be lowercase a here, and it's going to be lowercase b down here. And on this section, it's going to have uppercase A here. Sorry, that was meant to be red. Uppercase A here, and lowercase a here as it had before. And it's going to have now here uppercase B, and it's going to have lowercase b as before here. So I say, and that's a really good phrasing to use, that it creates new, just write it here, new combinations of alleles, because capital A used to go with capital B and lower a used to go with lower b, and now I've got a lower a with lower b here, and so they've exchanged places. So it creates new combinations of alleles. This phrasing basically comes up in a lot of mark schemes, so it's a good one to use. They will still have the same genes, so same genes at the same positions of the chromosome, but different alleles. What about independent assortment or segregation, and how does that help with, or contribute rather, sorry, to genetic variation? First of all, how do we define it? It's the random distribution of homologous chromosomes along the cell equator in metaphase 1, which affects which chromosomes, and therefore alleles, are going to end up in different daughter cells. Again, try to copy that phrasing, maybe not word for word, but making sure that you include all of the keywords that you need. And this random distribution of sister chromatids in metaphase 2, which also will affect which alleles end up in daughter cells. Let's kind of like try to sketch that out so that we can understand what we mean. So again, let's say that we're starting off with a diploid cell here, 2N, and this diploid cell has, we're not going to draw 23 chromosomes that humans contain, but let's just see it through the example of 2 chromosomes. So here's 1 chromosome. Let me just do red and blue so it's super clear. So this diploid cell will contain 1 maternal, for instance, chromosome 1, and another chromosome 2. So this is chromosome 1, chromosome 2 from maternal, and it will also contain their homologous chromosomes. So paternal chromosome 1 and paternal chromosome 2. When the first meiotic division takes place, how could these chromosomes separate from each other? Well, one daughter cell could inherit, for instance, both maternal chromosomes. So we go chromosome 1, maternal, chromosome 2, maternal. And the other cell could inherit both paternal chromosomes. 1 paternal, 2 paternal. However, they could also be the other way around. So I could have and that will happen if maternal chromosome 1 is on this side of the equator compared to the other. What if those chromosomes line up differently in the equator of metaphase? Well, so I could then have here paternal chromosome on this side, and the other paternal chromosome number 2, can go on the other side. And equally, I can have my maternal chromosome 1 on this side and my maternal chromosome 2 on this side. So now how will these chromosomes separate into daughter cells? Well, now I'm going to have maternal chromosome 1 here with paternal chromosome 2 here. And now I'm going to have paternal chromosome 1 here, and I'm going to have maternal chromosome 2 here. So you can see that the daughter cells are not genetically identical. So the number of different egg or sperm that can be produced from a meiotic division are always going to be different. There's going to be genetic variation of them. I just want to make a really quick note that pay really close attention to whether the question is asking you, how does meiosis contribute to genetic variation? In which case you will talk about the random distribution of homologous chromosomes or independent assortment of homologous chromosomes and the independent assortment of sister chromatids in metaphase 2. If the question asks, how does sexual reproduction as a whole contribute to genetic variation, then you will also mention the fact that it's the random fertilization of gametes as well that contributes to genetic variation. Let's see a typical question on meiosis, and particularly on the how meiosis contributes to genetic variation. So crossing over and independent assortment are critical for introducing genetic diversity during meiosis. Describe the process of crossing over for 3 marks. So again, let's break this down. What is my context? It's meiosis and genetic diversity, the relationship between the two. What is my command word? It's my command word is describe, yes. But I'm not going to go into too much depth. I'm just going to basically describe or outline in a way the stages of crossing over and how it comes about. And finally, my direction is that I'm only focusing on crossing over and not independent assortment. So not independent assortment. So take a minute, it's 3 marks. So take a couple of minutes to see. Oops, sorry. I'm so sorry. Switched over to the answer page too quickly. Take a couple of minutes to outline the stages of crossing over. Let's have a look at the answer together and break it down and comment on it. So one thing that we can say about crossing over is when it occurs, right? So it occurs in meiosis 1. And it allows alleles to be swapped between homologous chromosomes. That's also kind of like close to how we would define crossing over, right? So that's 1 mark. The homologous chromosomes line up and wrap around each other. So now we're describing the actual process, which causes bits of each chromatid. You can say sections if you don't want to say bits. Sections of each chromatid to break off and exchange with each other. That's a 2nd mark. Effectively swapping alleles. That's my 2nd mark. The points where crossing over occurs are called chiasmata. Something that we didn't explicitly cover. The chi, for those of you that are familiar with the chi-square test, chi just means x in Greek. So they call them chiasmata because they literally look like they form an X as they're swapping the bits around, the sections around.